Welcome everyone. It's great to see so much interest in our OBIS webinar on genetic data. I think it's also shows that it's really necessary to have such a meeting, and I'm really also pleased that together with GBIF we have developed the standards now in Darwin Core that we can actually capture all the metadata that's necessary to understand the genetic data. And also there are so many new DNA projects being set up that there is clearly a need for OBIS to be able to manage this kind of data. So we will have a number of presentations, so 4 presentations, and then we will continue with the questions and answer session. I would suggest that we use the chat box to ask questions, and so I will collect all the questions and then we will answer them one by one. If we need more clarification, then I will ask the person to turn on their microphone and ask the questions to us. If not, then I would ask you all to maybe close your camera and mute yourself, so that we can use the maximum speed and the best audio also for the meeting. And I hope we can have this as a very fruitful and joint discussion. So without further ado, I ask Sarah, my colleague, to start the presentations. Thank you.
Hello, everyone. My name is Sarah Suominen, and I work for the Ocean Biodiversity Information System. I'm going to give you a very short overview of what is OBIS and how genetic data is being incorporated into our database. OBIS is a global marine biodiversity database. Our mission is to provide free, accessible, and detailed marine data for all. At the moment, we have about 79 million presence records in approximately 4,000 different data sets, comprised of about 156,000 different accepted species names. In this map, you can see the distribution of occurrences across the globe at the moment in OBIS. So how does OBIS do this? OBIS collects species occurrence data from many different sources. This can be, for example, tracking data, data from other specialized databases like fish databases or algae databases, and observational data from different institutions. We provide long-term sustainable storage of your biodiversity data. The data is stored in a machine-readable format with all relevant metadata, including measurements of environmental parameters. The data is stored so that it is reliable and can be compared across time and space, across the globe, across different institutions, and possibly also across different data types. The data is stored in a standard format. This means that it has universal and comparable language use, and it also has contextual information on how data was acquired, or in other words, metadata. For this, we use the Darwin Core archive. This is a community standard format comprised of different tables, the core tables and extension tables with metadata in XML files put together in a zip archive. So as a data provider, you would provide most of the information that is required to search and acquire your data in the core tables. And then these core tables can be added to with extension tables depending on the data type that you are working with. So what is special about genetic data? Or why are we now interested in adding genetic data to OBIS? Well, as we all know, probably, genetic data is becoming increasingly popular also in the biodiversity field. It has enormous potential to increase our access to biodiversity information. Genetic data can be in the format of environmental DNA studies, specimen DNA, or bulk sample DNA. The challenge that we have now is that we can share this data in an accessible and reliable manner for the public, for management purposes, for scientific studies, and so on. The good news is that already raw sequence data is shared by most studies in public databases like the NCBI. However, raw sequence data does not allow you to search for specific species, specific locations, or specific time points. It will always require a lot of analysis of the raw sequence data to be able to do these comparisons across data sets. However, now this is going to change, and in fact, analyzed data can be submitted to biodiversity databases like OBIS along with GBIF and ALA. And especially we are interested in a sequence data that is linked to spatial coordinates and time points. So how this is done through a new extension called the DNA-derived data extension. And this extension allows you to add sequences and key metadata on how your sequence information was acquired to OBIS. Now, first of all, if you are interested in adding your genetic data to OBIS or other biodiversity databases, GBIF has published a guide on how to do this called Publishing DNA-derived Data through Biodiversity Data Platforms. And we highly recommend looking into this. It has really detailed information on how to go about adding your data and what is the contextual information around doing this. So what makes genetic data so different from other occurrence data? By definition, DNA data is derived information. It is not direct observation from the environment. This means that there is extensive sample processing needed in the laboratory as well as on the computer in a bioinformatic workflow. The data is reliant on reference databases, and there will always be, well, at least for a long time still, there will be a lot of unknown sequences. So the extension is meant to allow recording how the work was done to achieve the results that are put into the data tables so that we can always come back to the data and understand what was done. So I'm just going to go over a couple of genetic data workflows to give you an example of what kind of metadata you can record in the Darwin Core archives. First of all, we'll look at eDNA metabarcoding, so environmental DNA metabarcoding. Here's a typical workflow and the different steps that you would take. So first of all, you have your field sampling, you extract your DNA, you amplify a biomarker, which is then sequenced. These raw sequences are submitted to a public repository, analyzed, and you get your reference sequences, your abundance tables, and your taxonomy tables. And these can then be submitted to biodiversity databases. The metadata linked to each of these steps could be, for example, the following. So in your field sampling, you have your date, your coordinates, possibly environmental measurements. Your DNA extraction, you would have a concentration, your extraction method, the extracted amounts. Your biomarker, of course, you have primers, what is the target gene, the conditions. For your raw sequences, you know your sequencing platform, you will have an ID from the public repository. And of course, the most important, the last part, what you will have from your analysis is the taxonomy of your sequence, the pipeline that was used, what was your reference sequence database, and your annotation confidence. So to go through how you can add these things to the Darwin Core archive, first of all, in the occurrence core, so here you will see the actual fields that are available in the Darwin Core archive. So in the occurrence core, you would add the main things of the field sampling, for example, the event date, your decimal latitude and longitude, a link to a possible sampling protocol that you may have stored in another location like protocols.io. You can link your associated sequences, so you can add the ID of the public repository where you have submitted your raw sequences. And of course, you will have information on the taxonomy that you have acquired from your analysis. So the scientific name, the organism quantity would be your number of reads. The sample size value would be the total number of reads in each sample. And in identification remarks, you can add, for example, a link to your pipeline version that you have used. So, but what the DNA-derived data extension allows is to add more detailed information on all of these steps. Specifically, there's a possibility to add more information on the DNA extraction and the PCR conditions, the PCR primers, the target gene. You can also add from your sequencing method, your library layouts. And then, of course, most importantly, you can add your actual DNA sequence, as well as the methods that were used for the annotation and the reference database that you have used. As a second example, we'll look at how this would work with a quantitative PCR study. Of course, this is very different because we are not acquiring actual DNA sequences. However, this is also going to become more and more important for monitoring purposes, because you can look at target species, for example, invasive species or rare species or endangered species in specific environments. So the workflow is slightly more simple. What you have is your field sampling, your DNA extraction, and then an amplification from this where you will record the quantity of your target gene. The metadata linked to this, similarly for field sampling and DNA, you have your date, coordinates, your concentration and method. For the amplification, you have a protocol. And for your target region, you know the species, of course, you know the gene region, and you know which primers you have used. And then finally, you get your quantity, so the concentration, as well as some quality information on your assay. So what fields are available in the Darwin Core archive files for this? Well, occurrence score is the same as was with the environmental DNA metabarcoding example. You have information for your field study, information from your taxonomy, so your target species, and the quantity of what you are studying. And then there are some extra fields in the DNA-derived data which may be interesting here. For example, you have for your PCR protocol, you can add information on your probe reporter and probe quencher. For the quality, of course, you can add information on your threshold quantification cycle and automatic baseline value, and your limits of detection, and which software you have used for your analysis. So as you can see, there's a lot of information you can add, and I know this has been also a lot of information to show in a short period of time. I hope I have convinced you that it is possible to add enough information to be able to understand what was done in each study through the Darwin Core archives. The extension is designed to be flexible enough so that you can add as much information as you want into your data sets. And we do recommend adding as much information as possible. However, the most important thing, of course, is to publish. So a simple good data set may have your DNA sequences linked to a location and time with a species annotation, as well as information on the target gene, your reference database, and so on. And then a link to a publication where you can find more information on what exactly was done and how it was done and for what purpose. However, the most importantly, recording your sequences into the OBIS, this will enable a lot of future work on biodiversity and the condition of our marine environments, because we can always come back to the sequence. Possibly we have been able to understand more about the taxonomy of that sequence with time, or we have more sequences accumulating in the database that we can compare to and see the dynamics of what is happening with that specific biomarker. How OBIS differs from other biodiversity databases is that we have one taxonomic backbone, which is the World Register of Marine Species, or WORMS. This means that we want scientific names that are found in this database. Now, of course, we know that taxonomy with genetic data is quite a messy business. So what we want is that all sequences are indeed recorded in OBIS, but then that's the name that you acquire from your database, if it is not an accepted scientific name in the WORMS database, to record it either as a higher rank, which would be available in worms, or as if it is a completely unknown species or a completely unknown name, to record the sequence as the highest rank available biota. And then more information can be added in, for example, identification remarks field. So you can still link your OTU ID or your taxonomy ID from NCBI, which might be like a phototrophic eukaryote, for example. In addition, I want to mention here that OBIS also accepts flexibly environmental information in the extended measurement or FACT extension. And if you are interested in how you can link your environmental data to your biodiversity data, I recommend looking at the OBIS manual on our website, and you can contact us also if you have any questions. And just a brief overview. So OBIS works with thematic and regional nodes as our data providers. So if you are interested in adding your data to OBIS, I recommend contacting your closest node. And the data is accessed through third-party applications or a data mapper on our website. And we will hear shortly more about how genetic data can be accessed through these access points. So thank you. That was it for my introduction. Next, we're going to hear more about the first ever environmental DNA data sets in OBIS from OBIS USA. Thank you very much.
Hello, everyone. My name is Diana Lascala-Grunwald, and I am a data scientist at CENCUS, the Central and Northern California Ocean Observing System. I also do some data management for the Central California MBON. And I'm going to talk today a little bit about using the DNA-derived data extension to submit eDNA data to OBIS. So before I get started, I just want to highlight some key players. In particular, Francisco Chavez and Katie Pitts, who are researchers at the Monterey Bay Aquarium Research Institute. They are the data creators for this data set, and they were my primary points of contact. In addition, I'd like to thank Sarah and Peter for their guidance in using the extension, and Abby Benson for helping me submit it to OBIS. So just getting started, I want to tell you a little bit about where these data came from. So the Marine Biodiversity Observation Network, or MBON, is a global initiative that integrates data from existing long-term programs. As a member of a IUS regional association, I kind of think about this as an initiative that expands ocean observing into biological and ecological measurements. The US MBON projects are sponsored by IUS and other agencies, and they integrate old and new observations with a focus on emerging technologies. So in this case, eDNA. These data are part of the Central California MBON project, collected by Francisco Chavez and colleagues, as I mentioned, and integrated in collaboration with CENCUS. The data themselves are part of the Monterey Bay time series. This time series is an ongoing program where measurements are taken monthly at 3 different locations in the Monterey Bay. Filtered seawater began being collected for other purposes and frozen in the mid-2000s, and water has been collected specifically for eDNA research starting in the mid-2010s. The samples I've been working with are filtered seawater samples from a single station C1, which you can see pictured here. So we chose a small subset of these samples for this initial submission to OBIS. So I used 60 water samples that were collected in 2006 and between 2013 and 2016. These samples underwent metabarcoding in the 18SV9 region, and then they went through an extensive bioinformatics pipeline, which I'll talk a little bit more about in a minute. Some of these samples contributed to a recent publication, so you can refer to that for a little bit more information. So I want to talk a little bit more about the role of bioinformatics in identifying taxa in eDNA research. So my background is in marine ecology. So when I think about identifying an organism, I kind of picture a diver who sees a fish and knows what species it is to some degree of certainty. But when you just have sequences to deal with, things are a little bit different. So in eDNA research, bioinformatics software is used to quality check the data and then identify these proxies for species or proxies for taxa. In this case, amplicon sequence variants or ASVs. Then they use software to assign taxonomy to those ASVs using a genetic database. So I think the important thing to know here is identification is a little bit more complicated. It's evolving. These genetic databases are growing. They're becoming more accurate through time. And these processes vary between research groups, and that variation can have an impact on how the data are interpreted. So to capture this complexity and ensure that samples can be interpreted in a sensible way in the future, Chavez and colleagues keep extensive metadata records. So here you just have the first few rows and columns of one of their metadata tables. I know you can't read it. It's just to give you a sense of the extent of this metadata tracking. And this table documents everything from details about the sampling campaign where the water samples were collected to ancillary measurements like temperature to where the sample was sequenced. In addition to this, these researchers publicly share their wet lab protocols on protocols.io, and they also share their bioinformatics pipeline, their code for that processing on GitHub. So in addition to all of this information, I was given 3 tables when I was initially asked to structure these data for submission to OBIS. The first was the metadata table that I just showed you the top of, which included metadata on collection, amplification, sequencing, etc. I also received an ASV table, which includes that ASV sequence, sort of a proxy for species or a taxon, and the number of times the sequence was observed in the water sample, aka the number of reads. And then finally, I received a taxonomy table, which matches each of those ASVs to their taxonomy kingdom through species. And then, as you know, my job was to match as much of this information as possible to Darwin Core Occurrence Core and to the new DNA-derived data extension. Fortunately, GBIF recently wrote a guide to publishing DNA-derived data through biodiversity data platforms. And I think Sarah may have already talked a little bit about this. So mostly what I want to say is that I adhered to their guidance pretty closely. There's a section at the very end called data mapping that really lays out what all the extension terms mean and what the key ones are for you to use. So I don't want to reiterate what's just in the guide or what's in the extension. Instead, I want to give a quick overview and kind of highlight some confusing points or points where I interpreted things a little bit differently. So to do that, I'm going to start by talking about mapping eDNA data to the Darwin Core Occurrence Core, and then I'm going to wrap back around to the extension. So first, I want to highlight some occurrence core terms that maybe have a little bit of a different interpretation in the context of eDNA data. The framework here is that we defined the event as the water sample and the occurrence as an observation of a particular ASV. So within that framework, organism quantity and organism quantity type has a little bit of a different interpretation. So normally I think about this as the number of organisms that was observed or the abundance or density, but here it's the number of reads for a particular ASV inside a water sample. Similarly, sample size value, sample size unit, I usually think about these as referring to number of water samples or transects or something similar. Here, it actually refers to the total number of reads in the water sample across all of the ASVs. So for the other data managers out there, you basically group by event ID and sum across the counts of all of the ASVs. Finally, I want to note that basis of record has a different controlled vocabulary term here, and that is material sample. Next, I want to talk a little bit about terms with flexible interpretations. So first, identification remarks, which is sort of a free text field where you can include comments or notes about the identification. In this field, I included the species designation from the original data along with the GenBank version where this name was obtained. And actually, the data providers ended up adding the GenBank version to their internal metadata tracking as a result of this. I'll wrap back around to why the species designation is important in the next couple of slides. For identification references, the definition was a list of references used in the identification. I put in that field links to the published paper, as well as a link to the GitHub repository where the bioinformatics pipeline code is stored. Material sample ID I found to be a little bit confusing. It was defined as an identifier for the material sample as opposed to a particular digital record of the material sample. But the problem was the language in the guide is a little unclear because the next sentence refers to biosample ID, which kind of confuses it with associated sequences. After talking with Sarah and Peter, we decided that material sample ID should be the ID of the physical environmental sample. So in this case, the water sample, if it was preserved and frozen somewhere. And then finally, associated sequences, which should be a list of identifiers of genetic sequence information associated with the occurrence. This is where I put the NCBI bioproject ID from the sequence read archive. So basically the location where the actual raw sequence data could be obtained online. Possibly unsurprisingly, the most complex part of this process was dealing with taxonomy. So we're going to talk a little bit about adventures in taxonomy. The first thing to note is just the NCBI GenBank draws taxonomy from NCBI taxonomy database. And this database is not a taxonomic authority. There isn't anybody keeping careful track and making sure all of the species names are up to date and various things like that. Worms, the World Register of Marine Species, is a taxonomic authority, but it is based on traditional Linnaean scientific names. It's worth noting that GBIF accepts some non-Linnaean species designations like bold, BINs, and unite IDs, but OBIS uses worms as its taxonomic backbone, and it will flag records that don't match on worms. In addition, many organisms in NCBI taxonomy database do not have a Linnaean scientific name, or the name is in dispute. So here I've given an example from my dataset, eukaryotic clone OLI 11007. It's clearly not a Linnaean taxonomic name. And then there's a number of cases when there is a Linnaean name, but worms and NCBI taxonomy does not agree. So a simple example of this is that I encountered regularly was eukaryota in the kingdom field and no other taxonomic information. Something that was a little more complicated, I'm going to give an example of, and I'm going to mispronounce the species name, but it's Hemistasia phaeocisticola or something. And you can see here that I've given sort of the NCBI taxonomy for this organism and the worms taxonomy for this organism. And you can see that although both of them contained the genus name, all the other names don't quite match up. And this is maybe not the worst possible example that you could imagine, but it did kind of raise the concern that one might be able to match a species name on worms, but with a completely different taxonomy associated with it. So here I'm going to talk a little bit about my solutions to those problems. And I've got solutions in quotes because they're definitely just a starting point. So for this issue that many organisms in the taxonomy database do not have a Linnaean name, I basically tried to filter all of those out and replace them with missing values. And I did this in a really simple way. I just filtered out any name that had more than 2 words in it. This data set was small enough that I could actually look at the list of names I filtered out and the list of names I'd kept and kind of skim it and make sure I hadn't made a bad mistake. But we're going to have to come up with something better in the future because that won't work for really large data sets. Once I'd filtered those out and replaced them with missing values, I wrote some code that finds the lowest rank scientific name that is a valid Linnaean name according to worms. And that's the name that I put in the scientific name column. So essentially, I wrote some code that checks the species name, and if it doesn't match, It moves on to the family name and checks the family name. If that doesn't match, it moves on to the class name, etc., all the way up to kingdom. Now for the second problem. When there is a Linnaean name, worms and NCBI taxonomies do not always agree. So in the simple example I gave with eukaryota in the kingdom field, what I did there was actually replace eukaryota with a missing value in the kingdom field because it's not a kingdom. But then I put biota in the scientific name column. And that's kind of Worm's term for like something that is alive. It's really broad, but it lets you keep the record in the data set. For this slightly more complicated case of Hemistasia phaeocisticola, we used identification remarks and taxon concept ID to persist the original NCBI taxonomy identification into the OBIS record. So as I mentioned before, we used identification remarks to include that initial name. So for example, eukaryote clone OLI 11007, we included that in identification remarks, so it was preserved. Then I used the Python package bioentrée to query the NCBI taxonomy database and get their taxon ID for that specific name. And that's what we put in taxon concept ID. Finally, I put the worm's accepted name and scientific name, as I've mentioned, but I left the other taxonomic ranks as they came from NCBI taxonomy. I'm not sure if this is the most accurate way of doing things, but I think it is the best way of kind of preserving the original data. So that was a lot on the occurrence file. Mapping back around to the DNA-derived data extension, I actually found the extension to be reasonably straightforward. I think the biggest challenge in the extension is understanding what is relevant for those of us who don't have a background in eDNA. So I had just a couple of notes here. For example, SOP, which is defined as standard operating procedures used in assembly and/or annotation of genomes. What we put in this field were the two links to the protocols.io pages for the wet lab protocols and one link to the GitHub repo with the bioinformatics pipeline, and those links were all pipe delimited. I also want to highlight these three terms, ENV broad scale, ENV local scale, and ENV medium. These are all ENVO terms, so they're standardized descriptions for the environment where a sequence comes from, and they link into mixes, the minimum information for any sequence. So it's a nice way of harmonizing across a bunch of different standards. The last thing I want to say is I'm still learning a lot about what's going on in the extension. So for example, right now I'm looking at these OTU related terms and thinking about the differences between OTUs and ASVs, which is what I've been dealing with in my data set, and thinking about the best ways to capture the algorithms and thresholds for different species concepts in this bioinformatics pipeline that we've been talking about. And finally, I just want to say that there are many terms in the extension beyond those that are required or recommended in the guide. I just went with the basics this time around, just tried to hit those required and recommended fields, but there's a lot more you could do. And my best advice is if you don't have a background in eDNA, make friends with your data provider because they're the ones who have the best sense of what is important here. So just to wrap up, if you are curious what this Darwin Core archive looks like, you can check it out on OBIS and GBIF. I also want to highlight that this is really cool. It's awesome that we can contribute DNA-derived data to places like OBIS. I also want to say that the extensions required and recommended fields felt really well tuned to me. They did a good job of capturing the most important metadata without completely overwhelming somebody new to eDNA. I do foresee some challenges around this, in particular around uptake by labs. And particularly, I'm concerned about labs that may not have well-developed metadata tracking systems already in place. It's a big ask and a big lift to put all that infrastructure in place. And in addition, we need to continue encouraging people to share their protocols and share their code publicly. Another challenge, as I've already mentioned, is understanding the nuance in this field of research. So what do all those extension terms mean? And I think the best thing we can do here is start collecting and creating resources for each other, primers that can help us get caught up in this new field of research. And finally, there are some ongoing puzzles that I think the community is going to have to deal with collectively. One of them is obviously this misalignment between NCBI taxonomy database and worms. And one thing we might do there is, try to develop some sort of mapping between species names on NCBI and species names on worms, or at least develop some best practices about which one of those taxonomies should be prioritized and when. Another question is what to do with control data. Control data in this field is really important. It helps you figure out, how to interpret the data, what to do with the analyses, etc. But control data, they're not really occurrences. And so they don't fit well into the Darwin Core framework. So that's an open question. And finally, I think there's a scientific question about how these data can be combined with more traditional observations. How is a user on OBIS going to know that some of the occurrences are from DNA and some are from, let's say, a diver underwater? And how can you, combine those data types in a scientifically valid and useful way? So that's it for me. Thank you for your time. I really encourage you to get involved here. You can contact me. My email is on the slide. You can also join the conversation on GitHub. You can see that I've linked to our issues page where people are already kind of talking about some of these problems. And please stay tuned. I think we're hoping to get a use case up on GitHub where you can actually, look at the data and work through my code and, participate that way. And finally, I'm hoping to give a talk about this at Ocean Sciences next year. So I hope to see you then, and thanks.
Hello everyone, thanks for attending our webinar. My name is Peter Provoost and I'm the data manager at the OBIS Secretariat. In this presentation, I will show how you can access data shared using the DNA-derived data extension. This presentation will focus on the OBIS R package, but there's another option as well, which I will show at the end of the presentation. So for this presentation, I created a notebook, which is available at this URL. So before getting started, you will need to install the OBIS R package. OBIS is available from cron, but if you want to make use of the latest features, including access to DNA-derived data, it's best to install it from GitHub, which I'm doing in this first code block. So to find DNA data in OBIS, we can make use of the dataset function. So the dataset function allows you to search for dataset metadata records in the OBIS system. And this function has a parameter called hasExtensions. So if you pass the name of our DNA-derived data extension using that parameter, you will only get results that have sequence data. So if we do this, we get just one result for now, which is Diana's eDNA dataset. The metadata record also contains spatial extent, so I put this on a map here. Next, we can get some actual data. So to do this, we can use the occurrence function. And I'm going to pass the dataset identifier for the eDNA dataset. And I'm also going to set the extensions parameter. This will ensure that DNA records are included with the occurrence results. So if I do that, I get a table with around 65,000 occurrences with all the familiar Darwin Core occurrence terms. Then I did a little bit of data exploration. So I created a table here with all the phyla in my data set. With the number of species and records, just to get a feel for the taxonomic composition of our data. So for now, all the DNA records will be nested in a single column in our occurrence table. And to get them out of there into a separate table, we can use the unnest extension function. So here I'm going to pass the extension name. as well as some fields that I want to keep from the occurrence table. So when you do that, you get a new table with the fields that I specified, but also with all the columns from the DNA-derived data extension. So let's take a look at some properties of our sequence data. So this table shows the target gene, primers, and the number of records. And as you can see, all the records in our data set are 18S sequences with these two primers. I'm not a molecular biologist myself, but I played around with the data a little bit. But I'm sure that most of you will be able to do much more interesting things with the sequence data. So for the remainder of the notebook, I'm working with a small subset of the sequence data. So I'm just keeping phylum Ochrophytes and only the first 100 sequences. So until now, we have our sequences as just character strings in one of our columns. But to be able to make use of a number of bioinformatics packages, we need to convert those sequences to a DNA bin object. So that's what I'm doing in this code block here. And so that gives us a DNA bin with our 100 sequences, which appear to have different lengths. So next, I'm trying to align my sequences using this function. And that gives me another DNA bin object where all the sequences have the same length. And this is a visualization of the aligned sequences. And then finally, I use the A package to calculate a distance matrix and construct a tree. So that's it for the R package. So there's another option for getting DNA records from OBIS. So if you go to the mapper and you do a occurrence download, you will see these two tick boxes here. And if you check DNA-derived data here, the resulting zip file will include an extra file which has all the DNA-derived data records.
Hello everyone, I'm Ward Appletons. I'm the project manager of OBIS. And I think we have seen a number of very interesting presentations, so I won't take too much of your time, but I just want to end with a number of slides on our future plans. So OBIS is very closely involved in the United Nations Decade of Ocean Science for Sustainable Development. So one of the goals of OBIS is to protect marine biodiversity and preserve a healthy ocean for sustainable development. So we are an active partner in a number of decade actions. So we support a number of programs like Marine Life 2030 and OBON, the Ocean Biomolecular Observation Network. And we ourselves lead a number of eDNA projects. Like OBIS partners with the University of South Pacific to develop a marine invasive species monitoring program using eDNA. And we will launch a new project to organize eDNA expeditions in marine World Heritage sites together with UNESCO World Heritage Convention. This will be a citizen science driven project. So for these eDNA projects, we also need our own data management workflow and bioinformatics pipeline. So all this will be open source, so you will also be able to use those pipelines. And of course, the data will be freely available through OBIS. We've had the presentation from Peter on how you can access the data from OBIS. So we realize we still need to further develop and make it easier to search for genetic data in OBIS. One of our ideas is also to develop a sequence lookup service, so you can submit a sequence and then see if that sequence has already been reported by somebody else in OBIS. It's also important that we remain interoperable with other genetic databases. Of interest here is the TEDWIG task team that develop a mapping between the MIX-S and Darwin Core standards for genetic data. So now there's need for a broker tool to translate from Darwin Core to MIX and the other way around. We also find it very important that there is this principle to publish once, harvest many times. So we really hope that all the data can flow into multiple data systems, including GBIF and OBIS. We will also regularly check the taxonomic annotations against newly and updated information in genetic reference databases. So we will flag taxonomic inconsistencies and make suggestions. Like originally the name was species A, but according to the new information from the genetic reference databases, they suggested species B. So I think we also think it's important that the users can search for both the original name as well as the suggested name. We're very open to suggestions and to further develop OBIS and support your needs as well. So you can contact us by email, and you can also post issues in our project team GitHub repository. Thank you very much. I hope you enjoyed these presentations, and we hope we can have a fruitful discussion now. Thank you. All right, thank you. So these were the 4 presentations, and we already had a number of questions in the chat box. And some of them are answered already also in the chat box. So so there were a number of questions from Stéphane Pézan on how we're going to deal with different versions of data sets, especially when they use different pipelines. I'm not sure who wants to address this question, and I'm also not very sure about the answer to that question. But Peter, yeah.
Yeah, I replied that we have, of course, we have data set versioning thanks to the IPT that we're using. But I believe that that was not really an answer to Stéphane's question. So the question was more about, well, different data providers publishing the same data using different pipelines. And I'm not sure that we have a solution for this at the OBIS level. So yeah, for now, it would be up to the user to resolve duplicates. Of course, as mentioned in the chat, if we use globally unique persistent identifiers, that will be really helpful to identify these duplicate records. But apart from that, we don't really have a solution right now to completely avoid duplication.
Yeah, then there were a number of questions on how we're going to deal with the differences in taxonomy with NCBI and worms and using temporary names. So Diana, so you showed that you're using identification remarks to drop those names and use something in the taxonomic hierarchy of worms that matches that's the nearest match of your taxonomy. In the worst case, it's just biota. Does anybody else wants to comment on that? No. So I think the question was really answered that, yeah, at the moment we use identification remarks or there's also identification qualifier where you can mention when you're not entirely sure about your identification. Yeah, there was some chat about joining that week working groups to discuss these things. I would really encourage you to join those groups, and so there are some links in the chat box. That's really the place where we want to discuss these things, and then we can follow the recommendations as closely as possible. Yeah, so there was a bit of a chat on using material sample ID, and so Robert, maybe you can turn on your microphone and ask your question.
Yes, hey, thanks for taking my question. So Diana pointed out a number of sort of, I would say, best practices that she was using in, for material sample ID and that sort of thing. And I'm wondering about how do you maintain consistency over the long haul for people's use of, say, material sample ID or other variables? Do you try to get that back into the language or guidance for that term over time, or do you just let the community just decide? How do you govern that definitional framework?
I'm not sure I understand your question, Robert. Like, are you concerned
Diana,
like the interpretation of it will change over time?
right. So you interpreted it a certain way, and for
Right.
your particular use case, it worked well because that was what you decided. But I was wondering, how do you promulgate that to the entire community over time and make sure that other folks are adhering to the same manner? I mean, in the chat, it looks like there was a suggestion for parent material sample ID is now a proposed inclusion in Darwin Core, and the relationship between that and your water sample versus the actual sample that was used for the genetic piece. And I'm wondering, how do you govern that over time so that everyone's using it consistently? I guess that's my question.
Yeah, I mean, that's a great question, and the others might have a more sophisticated answer, but just, I guess one way is like telling people what I did. It was just the first one, so it's not like we all must do this now. But we can start here. And the other thing is just my experience with the rest of Darwin Core is that the interpretation of these terms do evolve through time, and that is somewhat of a concern, but I think hopefully pretty quickly the community will reach some kind of consensus on, this is what makes the most sense. And we can move forward with that. I don't know if anybody else.
That makes sense. Thanks, Diana.
All right, so when we had a question from John Nichols, but what does it actually mean, privileged ones harvest many times? Maybe I can take that question. So actually what we meant here, John, was that we encourage people to publish your data set on any platform like IPT and then have aggregators harvest it. It can be harvested by OBIS, by GBIF, maybe in the future by others as well. And that's actually really what was meant with it. Not really publishing new versions, although that's perfectly possible with IPT now as well. That clarifies your question. Chris Meyer, you had I think you had a very good question. I don't know the answer to that one about how people interpret organism quantity if different applicant runs need normalized for cross comparisons. Who wants to take on that question?
Maybe I can just comment on it. I don't know if it answers the question, but so organism quantity, you will add also the field organism quantity type, which will be sequence reads. And then a sample size value will be the total number of reads in your sample or across all of the ASVs or OTUs that you accepted. So then you can always go back to relative abundance in your sample through that. Is that what you meant, or did you have something else in mind?
No, that's great, Sarah. That's great. I think that it would also let us write some algorithms that we could also potentially pick based on that relative abundance to do, you know, rarefaction approaches too. I have to think about the math there, but probably could work.
Yeah. And I also agree with you, Chris, that we should be building our best practices and sharing our protocols, and so that they can develop into community practices. So Wayan Guan, I hope I pronounced your name correctly. You pose a question on, does the Nagoya protocol apply to ocean eDNA data? And actually it does. So especially if it's about samples that are taken within your economic exclusive economic zone, then the Nagoya protocol applies. It doesn't, I don't think it, well, it doesn't apply to areas beyond national jurisdiction. There is another discussion at the moment under the UN Law of the Sea Convention to regulate marine genetic resources in areas beyond national jurisdiction. But the Nagoya protocol is only applies to areas within national jurisdiction. But yeah, you need to have permission to even sample eDNA from water samples. Yeah, so Doc and Racing, you referred to the GBIF guideline. That's indeed one of the way we can propagate our best practice. I fully agree with that. And I mean, and it is discussed within the TEDWIC arena. Meredith, right? Yeah, so Meredith, so you're referring to temporary names, and there is, I can share the link in the chat box. There is a paper we published together with Worms on what should we do with temporary names. I mean, names that we think are new species but are not yet officially published. And so there it was proposed that those go into identification qualifier, but it was advised to use a unique identifier attached to the name, so that we then can easily trace them back and update the information when the name is officially published. So Claude, I think if I understand your question, we want to be able, we want to provide a service that you can also search for names that are reported in identification qualifier or identification remarks, so that you can find records that use these non-official names. I also mentioned in my presentation that when we recheck, do the taxonomic annotation over again, I mean, when we have the sequences in OBIS, we can do a blasting again against the reference databases, and we can actually update the taxonomy. Well, we don't want to update the original names that were provided. We don't want to change the data that people provided, but I think it would be good practice to add flags to the data and make suggestions, like if we run it through another pipeline or through we blast it again, now we get this taxonomic annotation. And then I think it's important that users can also search for the suggested annotations. I hope that answers your question, Claude. If not, let me know. Chris, maybe you can explain a bit your question on the CITES occurrences.
Yeah, so this has come up in our discussions about regulatory and reporting obligations for samples. Is if you discovered that one of your sequences hits a protected or CITES species, are you potentially in violation if you move that sample across borders, not knowing that it was there? And has there been discussions pertaining to that? Are you obligated to report that then as a potential flag? And I think we need to get those conversations going, because this is going to definitely be happening all the time. And you know, we're thinking about what we need to do here at the Smithsonian, certainly, because we're worried about, you know, if we start storing the samples and then somebody goes and does something with it, are we, you know, under some sort of custodial responsibility for those dynamics? And I can imagine, you know, for your World Heritage sites and all kinds of stuff, it's gonna, and I think it's better for us to get ahead of that before, in some way, this community really needs to think about what's going to happen there, because it will happen.
Do you know if CITES also applies to environmental DNA?
Well, I think that's, you know, we've been talking with folks about there's take and no take. You know, there's not an intent in the letter of the law, and at least here. So there's, but then we've also heard from general counsel that, you know, water's unregulated. Well, it's all pieces and parts, you know, so it's a pretty gray moment. But you can imagine, I mean, if OBIS is all of a sudden sourcing all these occurrences of protected or species, you know, it's pretty easy to do a query of give me a plot of all CITES species that were derived from DNA data just based on the scripts that you guys just showed. And then somebody could say, well, where are the permits for all these corals? Any coral, anybody who does eDNA over a coral reef is going to run into problems there.
Yeah. Okay, so we'll have to think about adding the permit to the records. So Thomas, Oregon, you have your doubts about using organism quantity, which may end up that people will misunderstand the information. Should we discuss this a bit further? Thomas, maybe you can explain a bit.
Yes, my concern is that while you may be able to separate out the records, you know, using the R script, you know, and just find those DNA records, for many of the people who are searching, you know, they may be just searching on taxon names and, you know, looking at abundances, for example, using organism quantity. And if so, that's going to probably give a very, you know, easily misunderstood result. And it seems like something like reads really deserves a separate category rather than being lumped in with a category that also includes number of individuals, for example.
So is this something that the DNA-derived data extension can handle, or should we look into the extended measurement effect extension for that? Or sorry, Diana, you wanted to react to this?
Yeah, I mean, and I may have this wrong, so correct me, but it sounds like this is sort of an issue around how these data are accessed through OBIS and combining different types of data and possibly doing that in a way that is not rigorous or not accurate. I think we've discussed, you know, having some kind of option as, you know, through the OBIS interface where you could choose whether you wanted to include DNA-derived data in your search or exclude it. So I don't know where that conversation is, but I know that idea has been raised. But I also think it's a larger question. When you're searching on OBIS generally, you might get an organism count, you might get a density, and are all those things necessarily directly comparable? And I imagine that people who use these combined biodiversity data sets have some practices for handling this that I might not be familiar with. But that's my 2 cents there. I do think it's a really important question.
Okay, thanks.
Johnny Gonjarla from our Indian OBIS node, you have a question on the sequence of publishing your data. Maybe Sarah, can you address that question?
Yeah, it's so you don't have to have them in the public database, but it is very, very highly recommended that you have your raw sequences in a public database before you submit to OBIS. And generally, this is the practice before you publish as well, because you usually at the moment journals are requiring that you have your raw sequences published in a public database. So we kind of assume that most people will be doing this. And we very, very highly recommend it, definitely.
Thanks. Robert, you have a question on who wants to answer that question on where can I find? I think if I answer the question, where can I find the exact definition and how I can use it, the term? Is that so there is a link? Maybe somebody can add a link to that big page where you can find the organ terms, but or did I misunderstand?
This is Robert. My question is really about, is there a place in Darwin Core where you can describe how you used a particular term like material sample ID, for example? Is there somewhere in the data set that you can put how you actually interpreted that term, especially when it's maybe open to broader interpretation?
Okay, that's a very good question. Who wants to answer that?
Hey, Ward, I can just say that, Darwin Core has IRI analogs for most of the terms. So if you want to be very explicit about what you mean and what you're putting in as values, you can use the Darwin Core IRI versions of the terms to be very sort of specific and use IRIs.
Excellent, thank you.
Yeah, Thomas, do you want to? Well, you answered one of the questions on the individual count and organism quantity. Do you want to specify it a bit more, or?
No, that's fine. Thank you. I appreciate that.
Thanks. All right.
Did you mean me? I'm the other Thomas.
Sorry.
Yeah, it was just related to the discussion about organism quantity, that it is there's an individual count, which actually means there was this many fish in this school, and then there's organism quantity with that always must go along with an organism quantity type, and that is a more open term where you can use, for example, the Brown-Blanquet scale for botanic surveys and percentage of biomass and other things. So if you consume data, then you should know the difference of those two. And it's a bit ambiguous because you can map individual count using organism quantity and then have organism quantity type as individuals. But you could also use it as we do with the DNA-derived data extension. It was just to clarify that.
Yeah. That's correct. So organism quantity is what we use for, I mean, we call it abundance, or we used to call it abundance. So it usually goes with the volume or an area in marine terms. Yeah, thanks, Abby. Yeah, quantity and quantity type should always be used together. Yeah, and we also started using our extended measurement effect extension in OBIS to capture these quantitative data. That's also a bit of a difference between GBIF and OBIS, although we also promote using the Darwin core terms in the core file. I don't see any new questions. Did I answer all questions? Or are there some unresolved questions that I missed?
I think we had a few earlier in the chats that we didn't yet discuss. Or did we answer these elsewhere? I think Stefan asked, for example, it is already a great step forward to include taxonomic annotations of genetic data in OBIS, but are there plans to include functional annotations in OBIS, or are there plans to develop a system that helps discover functional diversity?
Yeah, well, I think Peter responded in the chat box on that. So, but Peter, maybe you can also answer it.
Yeah, in short, we're planning to add functional annotations, working with the Worms's trait database, but there's nothing concrete yet.
Okay, did we miss another question, Sarah?
I guess not, no.
All right. Yeah. So Peter worked very hard to make it possible to download the extension data, not only through the R package but also through the mapper. Actually, today he also added the functionality that you can filter data in the mapper now when there are when data are when records do have information in the extensions. That's great. So we'll have to make a news item about this. So you're the first ones to hear about this. And so yeah, I'm really excited. I also want to thank our speakers and Diana and Abby to help us with publishing our first eDNA data set using the DNA-derived data extension. And I'm really looking forward to many more data sets. Well, there will be more data sets coming into our eDNA projects that OBIS is running, but I also hope we will get more data by other projects. And yeah, if you have further questions, please don't hesitate to email us, use our GitHub repository. And so let's work together on this. And without any further questions, I want to thank you all for your participation, for your questions, and I hope you hope you found this very interesting. Thank you very much. Have a good day, good evening, and see you again later. Bye.